get started on, I don't think we got to 31 today in class, so it should just be 27 and 28. I forget to keep track of the chapters sometimes. All right. Before we get started, anybody have any questions? Maybe from Mastering Bio, from lecture today, from any of the lectures? No? You just come here and assume I'm going to teach you everything you need to know? All right. Then let's get started. Your biology fact for the day: your brain cannot feel pain, which seems counterintuitive, right? The brain is where we process pain, but you don't actually have pain receptors in your brain, okay? You have pain receptors on your skull and on your scalp and things like that, so you can still get hit in the head, but you don't have pain receptors in your brain. So when they do any kind of brain surgery that requires direct contact to the brain, they don't actually have to put anesthesia in your brain, and you can be awake while they do it.
So is headaches just like a fake thing?
Headaches is a blood pressure thing, and so it's usually the blood around your brain or just on your head, and so it's triggering the receptors in your head, not specifically in your brain. So when they do brain surgery, you're awake the whole time. Partly because if they put you under and then do brain surgery, they can't tell if it's working, right? So if you watch like Grey's Anatomy or Scrubs or whatever, and they do a brain surgery and the person's just like sitting there wide awake, and you're like, this makes no sense. That's actually real. That's how they do that, which I think is pretty cool. Your study tip for the day: very simple, very concise, very practical. Use technology, right? We live in a golden age where technology is incredible, right? And it's all right there at our fingertips, but sometimes we forget to take advantage of it and we get caught in a rut of just doing the same thing over and over and over, right? I know it's like the fifth time I've said it, but Mastering Bio has way more options to it than you probably think. Flashcards, quizzes, study tips, tons of awesome things in Mastering Bio. And taking it outside of this class, most of your textbooks have a lot of extra resources to them. Khan Academy and YouTube videos, even just like Yahoo Answers. I know that feels almost like cheating, but I do it all the time. I have a physics class, and if I don't know how to do a problem, I go to Yahoo Answers and somebody word for word explains it to me. And so now I know how to do it, right? Obviously, I don't do that for every single problem. I won't learn anything. But if I don't know how to do it, use technology, okay? So just don't forget to try and branch out. More than anything, we all use technology. Branch out in how you use technology, okay? Some exciting stuff today. We can talk about bacteria and then protists. So bacteria, prokaryotic or eukaryotic? Prokaryotic, all of them. Okay. 3 common shapes of prokaryotic cells. It's been over like 2 years now since I took exam 4, but I'm like 95% certain that I remember a test question that was just, it was super simple, but it was just, what do bacilli bacteria look like? I don't remember which of the 3 it was, right? Fortunately, I had looked at this slide and memorized it well enough that I was like, I'm pretty sure it's the rod-shaped ones. But this is my warning to you guys: don't just glance over this slide once, okay? Know the 3 names: spherical, rod-shaped, and helical, and the 3 scientific names. I think that's pronounced cocci, I don't know, bacilli, spirilla, and spirochetes, okay? So know, like, if you saw spirilla, know that that is helical. Or if you saw bacilli, know that is rod-shaped, okay? What are we looking at here? Gram-positive bacteria, okay. Why do I care? First of all, what does gram-positive even mean? Okay, traps crystal violet. can be stained. Why do I care if it can be stained? Why do I care if it traps crystal violet?
It's more susceptible to being treated with antibiotics, right?
Okay. Can you elaborate? You're on the right track.
Essentially, it just doesn't have as strong of a defense system. It just has the one plasma membrane, and then the cell wall, which is your peptidoglycan layer, and then in the other one, it has another plasma membrane on top of that.
Perfect. So in a gram-positive bacteria, we just have to design an antibiotic that can penetrate the two layers, right? We just have to design something that can get through the peptidoglycan and the membrane, and we can kill this bacteria, right? Easy cheese. Okay. In comparison, gram-negative. What's gram-negative? Stains red. Why?
The crystal violet can't be absorbed into the peptidoglycan.
Yeah, so the peptidoglycan layer is the layer absorbing this crystal violet. But we have a carbohydrate polysaccharide layer up there, right? So the crystal violet doesn't get through, and it just kind of rinses away, and you end up with a reddish looking bacteria when you have stained it and look at it under a microscope. So why do we care? Why would we take the time to stain all these things?
Reveals which bacteria is more dangerous.
Yeah, it lets us know which kind of bacteria we're dealing with, right? Do we have the 3-layer bacteria or the 2-layer bacteria, right? Because if you've got some kind of bacterial infection and it's a gram-negative, then you're going to have to use a different antibiotic, right? A lower level antibiotic may not be effective. It may not get through all 3 layers. So you need to know which kind of bacteria you're dealing with in order to receive the right treatment or give the right treatment if you're the medical professional. Okay? So understand very well what gram-positive means, what gram-negative means, how that relates, to their layers and as far as staining with crystal violet, and then what that means as far as applications in the real world for medicine, right? Which kind of antibiotics will be effective and which ones won't. Does that make sense for everyone? You guys have questions with these? It's going to be a very boring session. Come on, guys. All right. Archaea are not bacteria, right? They are their own separate domain, okay? Archaea. They don't have their peptidoglycan, okay, so they'll be gram-negative, okay? In this case, it has almost nothing to do with what kind of membranes they have. They do not have that peptide. Is that what it is? Peptidoglycan, sorry. They don't have that peptidoglycan layer, and so they won't absorb the crystal violet, right? There are 3 kinds to concern ourselves with: methanogens, extreme halophiles, extreme thermophiles. What's a methanogen? Besides what it obviously says right there on the slide.
Wait, so these are 3 different types, or these aren't characteristics of every one of 3 different types?
Not all archaea will be all 3 at the same time. There probably are some archaea that are all 3 at the same time, but I don't know, of examples. So they'll have at least one of these. Well, I don't even know if they have to have one of those to qualify. Those are just 3 types that we look at. So, in your own words, tell me what a methanogen is.
So, are they the ones that kind of live in the bottom of the ocean or something? They absorb the methane, produce CO2?
Yeah, I mean, it doesn't have to specifically be the bottom of the ocean, but it's using hydrogen and carbon dioxide to make methane, right? So that portion where he showed us some hillbilly with no shirt on lighting stuff on fire in a swamp, right? I don't know if he was a hillbilly, but he wasn't wearing a shirt, so I assumed. They produce methane, right? And so he was able to light that methane on fire. It's natural gas, right? So this type of archaea produces natural gas, okay? Methanogen, think methane. Very simple. What about an extreme halophile? It is literally right there on the slide, yet it feels like we're pulling teeth.
He gave the example of like how they dump like a really salty solution out in the desert and let it dry, and it kind of grows these guys and turns different colors.
Yeah. So they can live in much higher salt concentrations than most organisms, right? You guys ever wondered why there are no fish in the Great Salt Lake? It's insanely salty. Fish just can't survive in that, right? All of their water would diffuse out of their body, and they would literally dry up, right? That's actually how salting things works, right? Have you ever, they used to do it more in the olden days, but you salt your meats and they last longer, dries them out, right? So fish would dry out. Most organisms would dry out in the Great Salt Lake. But there are little, almost plankton-like creatures in the Great Salt Lake, okay, that can survive in these really, really salty environments. Okay, so I don't know where the word halophile comes from. It's probably like Latin or Greek for salt-loving, if I had to guess. So you'll just have to memorize halophile means salt-loving, and so they can live in extremely salty conditions. That makes sense? Not the most difficult concept in the world. And then extreme thermophiles. What's that?
They can survive in really, really hot, like boiling water.
Yes. Usually you can boil your water and you do kill all the bacteria, right? And you kill pretty much everything in there because most things cannot live at above boiling temperatures. These archaea can, right? Most of the time they won't do anything, right? So it doesn't mean that you should stop boiling your water because it's not going to work anyway. It still works. It's going to kill pretty much all the harmful stuff. But this is a thermal vent in, I want to say Yellowstone, if I remember right. I don't remember if he said it in class. And if they take samples out of there, there should be nothing in there, right? It's way above boiling. Okay, these things are insanely hot. But they find little archaea in there. Or in the thermal vents at the bottom of the ocean that gush out super insanely hot water straight from the core of the earth, right? Well, the water doesn't come out of the core. The lava comes out of the core. And then, super crazy hot water. And they take samples around that and they find archaea, right? They can survive in these super, super hot temperatures.
Are any of these archaea like harmful to people, like harmful to us?
I don't know. Yeah, I have no idea. It's a good question.
What are the purpose of these things? Like archaea?
What do they do?
What do they do?
Eat, poop, breed? I don't know. Same purpose as kind of all organisms. They're just there. They're just living their life. I don't know. I know we can use methanogens. If you're asking like what can we do with them, sometimes you can use methanogens for breaking down waste and things like that. They'll eat it and then produce natural gas, so we can get natural gas from them. The thermophiles and halophiles, I think they're just chilling. I don't know that much about them, to be honest with you. I know what's on the slide. As terrible as that sounds, this is as deep as your knowledge needs to go on these, right? But it's a valid question. What do they do? I don't know. Other questions? Okay, so going back to bacteria, we needed to define the term pathogenic. What does that mean? Harmful, right? Disease causing. Okay, pathogens cause disease, right? So, it's I'm trying to think, less than, this pen, man, I don't know, less than 1%, oh my gosh. It's less than 1% of bacteria are pathogenic, right? So, he showed us that there's more bacterial cells in and on your body than your own body, right? So you would think we would just be sick nonstop and we would die when we're 10, right? We used to. Then medicine came around. but it really is a super small portion, right? Now remember, less than 1% of bajillions of bacteria is still a lot, right? There's still quite a few of these. But you don't need to like be using hand sanitizer every 10 minutes to try and kill them. That would make it worse, because then you would kill all the good bacteria, and you would keep all the opportunities away from your body to learn how to protect itself. And so then you would get sick. You get sick more often if you over sanitize than if you don't sanitize. So people who are like intense germaphobes get sick way more often than normal people, which is a little ironic, but I don't think they're doing it on purpose. It's a legitimate phobia. They're not like, I want to do this on purpose. Okay, anyway, a little bit of a sidetrack, but that's my little public announcement. What do you call those? Public safety announcement. Don't over sanitize. Also, don't like under sanitize. Just go out and eat dirt. Okay, exotoxic and endotoxic bacteria. Sound what they are. Exo, outside, endo, inside. It just refers to these pathogenic bacteria either secrete their toxins outside of their own cell body, or they hold it inside themselves, and then, when they finish their cell life cycle, they die, they burst open, and then their toxins get out and infect whatever organism they're in or on, right? Which ones are more dangerous? Exotoxic. You guys remember why?
Always putting stuff out.
Yeah. Even if this bacteria lives 10 minutes, it can put out a lot of toxins in those 10 minutes. Whereas an endotoxic bacteria that lives the same amount of time can really only put out toxin once, right? When it dies. They're both dangerous, right? They can both cause illness, but you should worry more about exotoxic. Okay. Why else should you care about whether or not a bacteria is exo or endotoxic?
Usually determines the type of wall that it has.
Usually depends on the wall that it has.
Right, so you can kill the exotonic ones are harder to kill.
Exotoxic are harder to kill.
Toxic, yeah, because they have 3 layers instead of just 2.
Okay. So 1 point, they're harder to kill, the exotoxic. 3 layers. What else? Think real hard. Why would I care whether or not it's exotoxic or endotoxic?
Maybe the way you treat it.
Okay, elaborate.
Just how you try to solve the problem or cure it, I guess. I don't know.
Yeah, you're on the right track.
Well, I mean, like if it's endotoxic, then you just want to get rid of it instead of killing it.
And if it's endotoxic,
Well, I mean, I don't know.
wouldn't you be accelerating the disease if you started killing all the endotoxic bacteria right off the bat? Wouldn't you kill all of them at once and have a whole bunch of toxin all at once? Wouldn't it be better to neutralize those bacteria and then kill them? So that's why it's important too. You have to know. If I go in and just kill all these bacteria, am I going to solve the problem and get rid of all these exotoxic bacteria? Or am I going to accelerate the sickness by helping the endotoxic bacteria spread their stuff faster? Okay, so you can design and use drugs that would neutralize them and then kill them. Okay, so these are why these things are important in medicine. I know you're probably sitting there thinking this isn't going to be like on a test. It could be, first of all, maybe not that deep of a question, but for those of you that are in any kind of biology major or chemistry major, whatever professional career you go into, you're going to have to care about stuff like this and know stuff like this, right? So this may come up 5 years from now, but understand why it matters. Sorry, questions on that before I move on? All right. So, why don't we just dump in the strongest antibiotic every time you get the sniffles?
Because the bacteria actually can evolve to become resistant to whatever you're trying to battle it with.
Yeah. Perfect. So if you just kill every bacteria with the strongest antibiotic every time, eventually you're going to teach those bacteria to resist the strongest antibiotic out there, and now you can't kill them, right? Also, the sniffles are caused by a virus, so you couldn't do it anyway, but it's a valid
Worried
point.
about it getting so far that they wouldn't be able to treat it, or not really? Like with antibiotics, that it would evolve.
Well, so how many of you have ever taken penicillin? A few of you. Do you remember what you took it for? I took it for acne, right? Just in high school. I was a super pubescent teenager, and right? Penicillin can treat stuff like that. Have any of you ever taken penicillin for a bacterial infection? Probably not.
I think I took it when I got my wisdom teeth out.
You took penicillin, okay. More of as a preventative measure, right? Yeah. So that keeps some of the bacteria away. Penicillin, in a lot of cases, doesn't work anymore, right? Because it got invented forever ago. I don't know how long ago. And all the bacteria evolved past it, and penicillin just doesn't work anymore. So we're constantly having to come up with new antibiotics. They don't necessarily have to be stronger. They can be. They have to take a different route, maybe. Maybe they have to break down the cell wall, or they have to neutralize the toxins, or there's a lot of ways that they can do it. But yeah, bacteria are always evolving, so we're always having to try and figure out different ways of getting around them and taking care of them. And actually, penicillin is starting to become effective again, because they've so far evolved away from penicillin resistance that they're starting to become susceptible to penicillin again, and they're starting to think it may cycle, and so we may actually be able to go back to really old medicines and kind of recycle through them, but I wouldn't be that optimistic. They'll just learn how to resist both, if you want my opinion. But yes, so they have large numbers, right? We know that. Bacteria, there's billions of them on a single person, right? More than that. They have short generation times. What does that mean? Yeah, they die and reproduce like madmen, right? Way faster than rabbits, right? And they have rapid evolution by mutation, right? So they evolve constantly, okay? So I have a video here. I have two purposes with this video. One, I think it's a great demonstration on how bacteria do this. And two, I like to show it to people who tell me that evolution isn't real.
Is this the large petri dish?
This is, yes, it's called a megaplate. They did this at Harvard, so you know it's...
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. After about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
11 days. And they were able to resist 1,000 times the normal antibiotic level, right? So for people that come to you and say, well, I don't know, I don't think evolution's real. There's no way we evolved from monkeys. First of all, evolution never said that. They say we have a common ancestor. Second of all, you literally just watched evolution in 11 days. So it's real. I just like pointing that out.
So how do they
somebody got a bacterial infection, like these MRSA superbugs and stuff that are already going around, how do they like treat?
Super, super, super strong antibiotics. And if you had to, they could isolate you so that you would be in a clean room, right? And the only thing your body would have to focus on is fighting this infection. So they'd pump you full of super crazy strong antibiotics, put you in the clean room, and you would just fight this infection. But that's for like quarantine level, Walking Dead type, like end of the world, super crazy. It's not something that living in Logan, Utah is ever going to happen to
Is
someone.
that harmful to like your own microbiome too?
How do you mean?
Like the other bacteria that are just helpful ones that are living in there?
I'm sure they would also attack your own bacteria. They'd attack everything, and it would not be fun. I can tell you that much. Yeah. So that's why bacteria can be tricky, right? You got to use the right antibiotic in the right dosage. If you do too strong, you may just be making them stronger for the next time it comes around, right? People who are prone to infections have to be really careful how they do this, because if you get an infection and you fight it with something super strong, but you're pretty prone to infections, you're going to get another one, and it's going to come back stronger, right? So they have to be careful with how they treat these. There is actually a lot of science behind treatment of bacteria, right? You go to the doctor and they just write something down, and you run to the pharmacy and you take it and you're good. But that doctor spent a long time learning how to treat what with what, right? There's a lot of science behind this. And I think it's fascinating. I love that video. I think it's crazy. Because you watch it and you're like, man, this is nuts. And then he says 11 days and you're like, holy crap. So, yeah. That's why you have to take all your antibiotics. That's why they usually go for 10 days, right? You got to get all of them. Yeah. Anyway, if you get on YouTube and Google megaplate evolution, Harvard, you can find that video and you can show it to your non-believer friends who are like, evolution's not real. Yes, it is. We won't get into the religious discussion of all of the rest of it, but evolution's real. It's not disproving anything else. But all right. Let's talk about metabolic diversity. Super crazy fun stuff. I know, it's fascinating. 4 kinds. Well, 4 kinds right here. You have, their energy source and their carbon source. 2 kinds of energy sources and 2 kinds of carbon sources. So phototrophic is what it sounds like. Photo, light. They get their energy from light. Chemotrophs get their energy from chemicals, right? So which are we? We're chemotrophs, right? How do we get energy from chemicals? That sounds like you drink Windex. Food, right? Food's made of chemicals, just like everything else. We hear chemicals and we think, chemicals, they're so bad for you. There's chemicals in carrots, right? Like everything's made of, not everything. Food is made of chemicals, right? So we get our energy from external chemical sources, right? Plants, things like that, from light. Now, what do they do with this energy? What do we do with it? Why do we eat? Besides that steak is really good.
Get energy.
To get energy, why? What do I want energy for?
Don't die.
Don't die, got to run our cellular processes. What else? Tons of stuff, but there's kind of the hint up there on the board.
Get carbon?
What are we made of?
Carbon.
Carbon. So if I don't eat food, or if a plant doesn't absorb light, we don't have energy, right? And we use that energy to fix carbon. All of our cells have carbon in them, right? Everything in this room has a little bit of carbon in it, right? Maybe not everything. The air probably, hopefully, doesn't. Well, it has a little bit. Carbon dioxide, because of me, because I talk the whole time. So we use the energy to fix carbon. We use it for tons of stuff, but one of the things is to fix carbon, right? So that I can replace skin cells and bone cells and liver cells, right? We got to get carbon. Okay, and plants have to do the same. And everything, all living organisms. That's why they call it carbon-based life forms. Everything living on this earth has carbon. Okay, questions up to there? Do you guys understand? I guess I didn't really go over. Autotrophs use atmospheric carbon dioxide, right? So they can just suck in carbon dioxide. Who does this? Plants, right? That's why we have such a wonderful relationship with plants. We breathe out carbon dioxide and they suck it up. And then they breathe out oxygen and we suck it up, right? We go back and forth. You guys ever heard that talking to your plants is good for them? It's actually true, but it's not because you're building their self-esteem. You're puffing carbon dioxide onto them. You're saying, oh, you're such a cute little good plant. Puff, 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 carbon dioxide. It's true. It looks like I'm vaping or something, but that's how it works. You breathe carbon dioxide onto them, and they use it to build their cells and run their processes, right? Doesn't work the same if you put your face right next to somebody else and puff, 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 they'd pass out. But they would also feel very good about themselves. And then heterotrophs from organic nutrients, right? Organic just means contains carbon. So fruits and veggies and meat and dairy and everything on the nice little pyramid that the government gives us. Heterotrophs. Why heterotroph? Where does that word even come from? What does hetero mean? Opposite or different from or outside of, right? So we do not produce our own carbon, right? We get it from an external source, right? We can't just grab carbon out of the air and say, got it, carbon, right? We take it from someone else. Essentially, the plants are the autotrophs. They take the carbon out of the atmosphere, and then we come in and say, thanks for doing all hard work, and we take the carbon that they fixed out of the atmosphere. Okay? Not a fair trade, but we're better than plants, so. Questions up to there? Is everyone understanding these terms? You feel confident with them? Okay, let's make them more difficult. Now you have to combine them, right? Because you have to fix energy, but you also have to fix carbon, right? So you have 4 different combinations. Photo-auto, photo-hetero, chemo-hetero, and chemo-auto, right? So what's a phototroph again? Light energy, perfect. Autotrophs? Atmospheric carbon, right? So most plants, well, I think all plants, actually, no. Like Venus flytraps and stuff like that are photoautotrophs and, I want to say, chemoheterotrophs. Photoheterotrophs. I don't know. They're a weird one. Like Venus fly and pitcher plants and stuff, they're kind of an exception. But pretty much all plants, right? Absorb light energy and absorb atmospheric carbon, and that's how they sustain themselves, okay? Chemoheterotrophs. What's a chemotroph? Get energy from organic material, right? So heterotrophs, what do? Eat other things. They get their carbon also from organic material. So what's a chemoheterotroph? An example of? Humans, bears, fish, actually also fungi, right? Fungi dissolve other organisms and eat them, okay? And they do not do photosynthesis. That's actually how they first realized that fungi probably weren't plants. At first they were like, yeah, mushrooms are just plants. And then they were like, wait, they don't do photosynthesis. And they eat other organisms. So they are their own thing. Okay, so chemo, we already decided, is getting your energy from chemicals. Autotrophs, what do? Atmospheric carbon, right? So they're kind of a mix, right? They eat other organisms and absorb atmospheric carbon dioxide, right? Prokaryotes are the only ones that can do this. And then we have the photoheterotroph. It's very rare. Also only prokaryotes. But they use light to make ATP, like plants, but then they get their carbon from organic material. So they absorb light energy, but they don't fix atmospheric or carbon dioxide the way that plants do. So they're kind of their own thing too. Make sense to everyone? If you saw a test question related to something like this, would you feel confident enough? Because you have like 2 weeks. And one of those is Thanksgiving break, and no one's going to do anything during Thanksgiving break anyway, right? So it's like we have 1 week. You better do something over Thanksgiving break. You have a test right when you get back. The nitrogen cycle. As soon as Dr. Mott said you don't need to worry about this in class, I deleted this slide. And then I went, I'm going to put it back just so I can say, don't memorize this, don't study this, don't go nuts over this, don't try and nothing, right? I put it in there to tell you, don't do anything with this. Do not waste your time. Okay, there's so many other things that are going to be on exam 4. This is not one of them. Okay, and end of story. Let's talk about protists. Super incredibly, awesomely fascinating. Prokaryotic or eukaryotic? Eukaryotic, right? So many excellent domains. These are the names that Dr. Mott kept crossing off in class. So I'm literally only showing them to you to say, be familiar enough that if you saw one of these names, you could go, okay, we're talking about protists, right? And then be familiar with those sicknesses that he pointed out specifically in class. African sleeping sickness, Giardia, I'm blanking on. There was one other one, wasn't there? Does anybody remember? Want to help me out? African sleeping sickness, Giardia, malaria, I think salmonella got pointed out as well. So be familiar with those. If you saw, African sleeping sickness is caused by a bacteria, a protist, a fungus, a plant, an animal. Probably know that bacterial or African sleeping sickness isn't caused by plants or animals, but it's not a fungal infection, right? It's not a bacterial infection. It's protists, right? If you saw what causes Giardia, no, it's protists, right? It's not bacteria. Because if no one had ever said to me before Giardia is caused by protists, I would guess that it's either a bacteria or a virus, right? That's what most things are caused by, so that's the safest bet, right? So don't do that. Don't go in and think, well, most things are caused by bacteria, so it's got to be bacteria. Those are some of the special cases. And again, if it gets pointed out specifically in class as an example, that means you could see that specific example on the test, right? That makes sense? Yes? Catching my drift?
Do you think he's going to ask us specifically what kind of protist it is, or just if it is a protist based on? Because he had the scientific name, then he had a different name underneath those.
Right. I would err on the side of caution, and I would memorize the disease and which protist caused it. So I would know, and this is where I draw a blank on which ones cause what.
But not the domains.
No, so you won't need to know like it's a euglenozoa that causes it, it's a Strepinopila that causes it, but dinoflagellates, right? You'd have to know more of those kinds of names. And the ones that produce glass that I'm also drawing a blank on. Who remembers the glass ones? Diatoms, that's right. So be familiar with those kinds of names, diatoms, dinoflagellates, but don't worry about the names on this as much. Be familiar with them, that if you saw them, you could go, okay, I know what we're talking about. We're talking about a kind of protist. Be more familiar with the names given in class and the disease they're associated with. And that's about the extent of it.
I kind of got confused about and I'm going to pronounce this wrong, the Euglena protus. What it was, he was talking about how the chloroplast has a nucleus.
Oh yeah, I don't think I have that slide, but I can find it. What he was essentially trying to tell us is there's something weird about their chloroplasts, is that those chloroplasts have nuclei, which is not normal. Most do not. Let's see. PowerPoint slides. It's closer to the end. Maybe not. Here it is. This is what you're talking about? So essentially what he's trying to show us is how this arose. And I can't draw on this since it's just Google Chrome. So I'll get in the way of everything. So endosymbiosis, right? That's how mitochondria and chloroplasts got into our cells, right? Because they're separate from us, right? And so they did the normal endosymbiotic relationship where they moved into our cells and they said, this is nice and safe and warm, and I can give you energy back, right? What they think may have happened is separate endosymbiotic events that allowed some of the chloroplasts to have nuclei inside of them. It's not necessarily, no one knows for sure that's exactly how it happened because we weren't there millions of years ago or whatever. So really what I would understand from that is just, first of all, who it happened to, right? So this is the Euglenozoa, the euglenoids. And I would just know that they have chloroplasts with nuclei. You don't necessarily, and maybe know that it was by some endosymbiotic events. You don't need to know all the mechanisms behind it, when it happened, where it happened, how it happened, just that euglenoids have chloroplasts with nuclei. That's kind of the extent that I would expect to need to know. Does that help? Okay. Other questions? Those are my final slides, because as I said, not a whole lot going on with protists. It's going to get a lot more complicated on Friday when we start talking about fungi and plants and animals. Much more complex organisms. And what did I warn you about with Dr. Mott? He freaking loves plants, man. And as soon as we get to plants, you got to know like all kinds of different structures and functions and types, and plant biology gets nuts because Dr. Mott's like a pro and he wants you to be a pro too. So if you feel relaxed right now and you're like, protists aren't bad, wait till we start talking about flowers. Freaking A, man. So be ready for that. That's all I'm saying. And again, don't get lax over the Thanksgiving break thinking, no, it's just protists. Because I'm pretty sure we're going to get to plants either Friday or Monday. And then you go on break. And that's when I would take advantage, honestly, of that little lull. Use the Thanksgiving break as a chance to either catch up or get ahead and be real confident. Because plants, for me, was the hardest part of exam 4. It gets heavy.
Plants is exam 4?
I'm pretty sure.
We're not going to include flowers and stuff on this next in exam 3?
Didn't we already take exam 3?
I mean, 3. Okay, I'm sorry. I thought you were talking about the final. I got it mixed up in my mind.
No, so this test is up through like fungi, plants, and animals, I think is how far we go. But I'm not positive. But definitely plants is on there.
Yeah.
And again, you may sit there and go, plants was like mega easy, and I don't know what you're talking about because you're smarter than me, and that's fine. But for me, personally, plants was super hard. So I always just put the warning out there. it can be very difficult. So be prepared for that, and just use the break as a chance to get really, really good at plant biology. Yeah.
Do you know when your super session's going to be?
Mine's going to be Monday. I believe Hillary's is going to be Tuesday, like the day before the break. And then Chase will do one the Monday we get back. And then the test is Tuesday. Because I'm not allowed to hold a test or a super session within like 2 days of a test. So normally I would have held it Wednesday, but it's Thanksgiving break, so literally 0 people would show up, including me. So yeah, it's going to be Monday. Hopefully people show up. Some of you will be like, I'll figure it out on my own over Thanksgiving break. And that's fine. Do what you want. Yeah, Monday. Other questions? You're all so respectful and quiet and just wait. You can leave. No one's holding you here. All right, thanks for coming. Somebody bring me